Oh, hello, everyone. Welcome to my sessions on microbiology. I'm going to be talking to you today for several hours and tomorrow as well. And I have been a microbiologist for many years. I love this area, and I have to admit, it's certainly a large one, and there's lots of information. So I'm tried to scale it down to what I feel is most important. And we'll start first with mycology today. So as the starting points, of course, yeast are unicellular and they bud to produce daughter cells. Yeast can grow in a variety of different kind of media in 24 to 48 hours, and they appear much like bacterial colonies, as you see in the pictures below. A few of them will be orange or colored, such as Rhodotorula, and some will be quite mucoid, as shown on the bottom right. Also, there's been many taxonomic updates, and you're going to see, I do not know how boards will present these, but I will talk to you when we do have some of the taxonomic updates and relay those to you. The other type of mycology, besides yeast, of course, are the molds. Molds produce hyphae and conidia, or what's known as spores, like shown in the picture. They also grow very well on solid media and can grow from 3 to 21 days at 30 to 35 degrees, depending upon the genera of the mold. Most of the mold colonies are going to produce pigment, which can help us identify them. And also, I'll talk later about some of the taxonomic updates. Fungi are very hardy organisms. They are not difficult to grow or transport, so you do not have to have any special transport media. You just need to use sterile containers and transport at room temperature. There's, of course, numerous anatomic sites that are appropriate for fungal culture, and I've listed those on this slide. What are the most common fungal medias used? Well, by far the one that's been used most over the last few decades is Savarod's dextrose agar, known as SABS. It's an all-purpose fungal media that does not have any antibiotics. Because of that, bacteria can also grow on this media. It contains 2% glucose and a pH of 7. Because there is no antibiotics and bacteria can sometimes contaminate cultures, this agar is best used for the subculture of fungi and for identification, testing, and susceptibility workup preparation. Over on the right, on the bottom, is inhibitory mold agar. This is a selected and enriched medium. It contains antibiotics, chloramphenicol and gentamicin, that can inhibit bacterial growth. Because of this, it is a very good primary recovery media for pathogenic fungi. Another one that's important to remember is mycocell or mycobiotic agar. This is a very selective SABS medium that contains both chloramphenicol and cyclohexamide. It's used primarily to culture dermatophytes. That's molds that can cause infections on skin, hair, and nails. You shouldn't use mycocell as a primary plating medium because it contains cyclohexamide that can prevent the growth of some of the very important pathogenic fungi, such as Trichosporon, Candida tropicalis, and even Cryptococcus neoformans and gattii. And then finally, we have Brainhart infusion, BHI medium, with or without blood. This is used for primary recovery of fungi. You can add chloramphenicol and gentamicin to prevent bacterial growth. And since you can add blood to this media, it could be very helpful for the nurturing of some of the fastidious fungi, such as Histoplasma capsulatum. How about safety precautions? Safety precautions are very, very important these days because we're seeing a variety of, very serious organisms. So for mycology is a BSL2 level safety precautions. You should do, for mold cultures, as pictured, you should do all of your work in a BSL2 biosafety cabinet with HEPA filtration. Yeast cultures can be worked on out on the countertop. The personal protective equipment should include lab coats and gloves, and you should autoclave all your biohazardous waste. Another safety precautions shown on the right-hand bottom picture, you should always seal all of the plates used for mycology culture to prevent mold spores from disseminating into the laboratory. So all plates must be sealed with oxygen permeable tape. Using BSL-2 precautions, you will inoculate specimens onto media and streak for isolation. You then seal the plates with the permeable tape prior to incubation. Tubes or vials can also be used for culture, but most laboratories don't like to use tubes or vials because they're much harder to visualize the growth of molds and yeast. You hold fungal cultures at 30 degrees centigrade for 4 weeks. This very long length of incubation time is necessary to look for the slower growing systemic fungi. And then you screen these cultures periodically to see if anything is growing. How do you identify yeast? Well, for decades, we used biochemical reactions to identify yeast. But with all the recent taxonomic updates and also new emerging yeast pathogens, it's become really quite challenging to use biochemicals. So what are the newer methods and improved methods for yeast identification? They include MALDI-TOF and sequencing. MALDI-TOF is when you smear colonies onto a little target plate. You put it into the MALDI-TOF instrument. It's superheated by laser and converted into charged protein particles. So the particles, based on mass and charge, travel up a vacuum chamber to a detection plate to form identification peaks that can be analyzed and identify yeast to genus and species. It's really an amazing new method that's been in laboratories for about the past decade, and it's currently the most popular and precise method for the identification of yeast. It's very rapid. You get results in about 30 seconds, and it's relatively inexpensive. If you have some very, very unusual yeast and MALDI does not identify them, you can go to sequencing. But this is just really reserved for the most problematic of all yeast. Mold identification. For years, we used lactophenol cotton blue preps. You can see the picture down in the bottom. This is an adhesive tape preparation that where you put a drop